Welcome to the Lecture 1 of the Module 4, Molecular Biology of Animals. Animal evolution began in the ocean over 600 million years ago with tiny creatures that probably do not resemble any living organism today. Since then, animals have evolved into a highly diverse kingdom. The number of currently living species is estimated to be between 3 and 30 million. And scientists still discover more species as they explore ecosystems around the world. The animal classification system characterizes animals based on their anatomy, morphology, evolutionary history, features of embryological development, and genetic makeup. This classification scheme is constantly developing as new information about species arises. Human fascination with animal behavior probably extends back millions of years, perhaps even to times before features of embryological development and genetic makeup. This classification scheme is constantly developing as new information about species arises. Human fascination with animal behavior probably extends back millions of years, perhaps even to times before formation of human in the modern sense. Modern behavioral biology draws on work from the related but distant disciplines: ethology, which focuses on the behaviors of diverse organisms in their natural environment, and comparative psychology, which focuses largely on a few species studied in a lab setting. Tinbergen proposed 4 basic questions helpful in understanding any animal behavior. Causation. What causes the behavior? What triggers the behavior, and what body parts, functions, and molecules are involved in carrying it out? Example. Singing is triggered in zebra finches by social cues, such as the proximity of potential mate as well as the appropriate hormonal state. The ability to produce songs is influenced by male hormones and occurs mainly in male birds. Songs are produced when air flows from the air sacs in the bronch through an organ called the syrinx. Certain parts of the brain control song production and are well developed in male zebra finches. Second, development. Syrinx. Certain parts of the brain control song production and are well developed in male zebra finches. Second, development. How does the behavior develop? Is the behavior present early in life? Does it change over the course of the organism's lifetime? What experiences are necessary for its development? Example. Young male zebra finches first listen to the songs of nearby males of their species, particularly their fathers. Then, they start to practice singing. By adulthood, male zebra finches have learned to produce their own songs, which are unique but often have similarities to those of their fathers. Once a finch has perfected its song, the song remains fixed for life. Number 3. Function or a finch has perfected its song. And the song remains fixed for life. Number 3. Function or adaptive value. How does the behavior affect fitness? How does the behavior affect an organism's chances of survival and reproduction? Example. Singing helps male zebra finches attract mates, increasing the chances that they will reproduce. Singing is a part of an elaborate courtship ritual that entices the female to choose the male. Number 4. Phylogeny. How did the behavior evolve? How does the behavior compare to those of related species? Why might it have evolved as it did? Example. Almost all species of birds can make vocal sounds, but only those in the suborder Passerii are songbirds. Relative to the zebra finch, other songbird species differ in the timing of their listening and practicing phases, the plasticity of song over their lifetimes, the extent to which the song is similar among individuals of the species, and the way that singing is used. For example, for defense of territory or courtship of mates. Brain development, brain activity and behavior depend on both inherited and environmental influences, and there is increasing appreciation that social information can in turn impact brain gene expression and behavior. Behavior-related aspects include migration and circadian rhythm. Please feel free to leave your questions and comments in the forum, and we'll be happy to answer them. Good luck, and looking forward to seeing you soon. AJ. A number of methods are presently employed for genetic engineering of various animal species. Most of these were developed originally in mouse and Drosophila models and have only more recently been extended to other domesticated animals. Access to the germline of mammals can be obtained by one of the 5 methods. First, direct manipulation of the fertilized egg, followed by its implantation into the uterus. Second, manipulation of the sperm used to generate the zygote. Third, manipulation of early embryonic tissue in place. Fourth, the use of embryonic stem cell lines. And fifth, manipulation of cultured somatic cells, whose nuclei then can be transferred into the nucleated oocytes and thereby provide the genetic information required to produce a whole animal. The last 2 methods have the advantage of allowing cells containing the modification of interest to be selected prior to undertaking the expensive and lengthy process of generating animals. Usable embryonic stem cells are not available for all species of interest, however, and generation of embryos by nuclear transfer from somatic cells is a method of choice for genetic engineering and application of nearly genetically identical animals. There are 2 basic approaches presently in use for inserting DNA into vertebrate germline cells: transfection and infection with retrovirus vectors. A third approach, based on the use of mobile genetic elements, has been commonly used for insects. As such, transfection methods include: first, direct micro-injection of DNA into the cell nucleus. Second, electroporation, introduction of DNA through transient pores created by controlled electrical pulses. Third, use of polycotyns to neutralize charges of DNA and the cell surface that prevent efficient uptake of DNA. Fourth, lipofaction, or inclusive DNA in lipid vessels that enter a cell by membrane fusion, much in the manner of a virus. And fifth, sperm-mediated transfection, possibly in conjunction with intracytoplasmic sperm injection or electroporation. The manner of DNA introduction is a technical issue determined empirically for each system and makes little difference to the final outcome. When transfecting cultured somatic or embryonic stem cells, a selectable marker, such as the gene encoding phosphotransferase, is often included as part of the DNA to permit selection for its presence. Introduction of DNA into random sites in the germline is a mutagenic event that will affect any gene that happens to be or near the site of integration. The most obvious effect is the disruption of the integrity of a gene into which the insertion occurs. Since a large fraction of the mammalian genome is non-coding DNA derived from various kinds of silenced transposable elements, not all integration events will lead to gene inactivation. Direct DNA introduction can lead to numbers of integrated copies at multiple sites, leading to a risk of creating animals with a variety of genetic defects, which should be carefully screened for in the course of subsequent breeding. For example, one of the very first transgenic mouse lines generated, intended to contain an inserted active oncogene, also suffered a lesion that caused a severe recessive developmental limb defect. Even if expressed as desired, the genetic engineering itself can often have unexpected effects on the physiology of the engineered organism. Please feel free to leave your questions and comments in the forum, and we'll be happy to answer them. Good luck, and looking forward to seeing you soon. AJ.